stuff. And in talking about the respiratory system, we had made our way into talking about the gas exchange, and one of the laws that's very important to the exchange of gases for the lungs is Henry's law. Because in the structure we have the respiratory membrane, and that means a water film is present, okay? And the gases of oxygen and CO2, they are going to need to be able to cross this respiratory membrane. If you remember, the respiratory membrane is formed by the capillary, the blood vessel, and that alveoli, that alveolus, okay? And so it's that their basement membranes pretty much right there together. And it creates a liquid environment. And we've got to get the gases across. We did determine that CO2 was the gas that is more dissolvable. All right? Because CO2 in our system is toxic. Oxygen isn't. And we have to make sure that there is the ability to affect their movement. So oxygen, we know, if you guys remember, when we look at a red blood cell, as that red blood cell matures, it has formed those little 4 protein chains, the globins. Does everybody remember that? And in the middle of it is a heme group. Does everybody remember that? And attached to that heme group is an iron. And oxygen is able to bind to a heme group. So oxygen, because it binds to heme, and these are globin chains, we get our term hemoglobin. Now, when an oxygen is bound to it, we call it oxyhemoglobin, because oxygen is bound to this heme group. So, like I was saying before, because we will have 4 heme groups, because you would have 1, 2, 3, 4, because I have 4 globins. Does that make sense? So, having them 1, 2, 3, 4, if every single one of them had an oxygen bound to it, we say it would be 100% saturated, which is what we were talking about the other day. You guys kind of remember me mentioning that pulse ox, okay? And yes, they want it to be as close to 100 as possible. They start getting concerned at roughly 95%. That's letting you know how important it is for that oxygen level to be up in our blood, okay? And if it's 75%, that would mean 3 of the hemes would have oxygens. If it was 50%, 2 would have it. Do you see how they get the saturation rate? Now, there's one gas that will compete for this binding site. So on that heme group where the iron binds to it, the one gas that will compete for it is carbon monoxide. How do we get exposed to that? Huh? Okay, think about the exhaust of your car. Think about anytime like a propane, butane, other types of gases are burning. When you smoke something, note I said smoke something, okay, carbon monoxide is produced. And one of the things about carbon monoxide, it's poisonous. So you get too much of it, it doesn't release from these heme groups. And because it doesn't release from the heme group, oxygen cannot bind, does not get to the tissues, you die. This is why carbon monoxide poisoning is so dangerous, okay? When no oxygen is bound to those heme groups, we say that the blood is deoxyhemoglobin. D meaning without, okay? When we talk about the relationship between oxygenated, non-oxygenated, or oxyhemoglobin versus deoxyhemoglobin, it's called a dissociation curve. Now, your textbook will show a really cool picture of the this oxyhemoglobin dissociation curve, because it is letting us know how that hemoglobin molecule, and the term used, loads oxygen. Let's think about this for a second. That red blood cell that's going to be passing by that alveolus, where I've got that respiratory membrane, and if it's passing by that alveolus, the goal is hopefully it's picking up oxygen if needed. Okay, so everybody following me? So that red blood cell moving through the blood vessel that is in contact with that alveolus, if needed, oxygen would load on to that hemoglobin. Does that make sense? Okay, now if the oxygen loads onto that hemoglobin, it will then come back to the heart into the left side, leave by way of the aorta, from the aorta make its way to tissues. So what do you think that oxygen will do at the tissue level? If it loaded, it will unload. So these are the terms they use. Does that make sense? Okay, so this dissociation curve is representative of the picking up of oxygen and then the unloading of it at tissues. And that curve will be affected based upon the need of the tissues. So right now, the need of the tissues. Let's look at skeletal muscle, for example. Right now, in the gastrocnemius, where is that? In the calves. Who's needing more oxygen to their gastrocnemius? Y'all or me? Okay, so now the dissociation curve would be greater for my gastrocs than yours. Why? Yours are at rest, mine are moving. So we'd have to look at oxygen binding to their heme groups, and that gives us information about saturation, And our curve, okay, will let us know how that's occurring within the body. So your textbook, I think, will show this really cool picture of somebody at rest and somebody with exercise and kind of show a curve that's occurring, okay? Why would that use of oxygen be clinically significant?
You mean as in exercise?
Right. Think about it in use of exercise or versus someone who may be sitting.
Because oxygen is needed to power the glycolysis.
We need it to power. Now, you said glycolysis, but that's okay. Because if muscles are in use, they're going to make energy by way of glycolysis. Do you guys remember that? Where you only got 2 ATPs? But if those muscles are at rest, how many ATPs would they be making?
No. If you're at rest.
If you're at rest, they're going to go into the mitochondria, go through cellular respiration, and make 36 to 38 ATPs.
And this is during rest.
That would be during rest. When your muscles are in motion, especially, thinking skeletal muscle, okay? When those muscles are in motion, the oxygen that is being channeled, being carried to those muscles is going to be used most likely for that anaerobic fermentation versus aerobic when they're at rest. And in some cases, cells can use just anaerobic fermentation, okay? Sometimes we have cells that need all of it to be aerobic, okay? But the goal is that we get the oxygen to the tissues, because the tissues, they contain the cells, and the cells have a job to do. And the byproduct of all of that is CO2, my carbon dioxide. Carbon dioxide, the way that we are going to transport this gas that is toxic to us, okay? Well, carbon dioxide is not going to compete for a heme group. Carbon dioxide will attach to the globin. So, our hemoglobin molecule can carry CO2, but it really wants to focus on the oxygen. So what we're going to find is that a lot of the way that we are going to transport our CO2 is to either get it to attach. Do you guys remember the little globins that exist? Okay? And if we're looking at a blood vessel, okay, globins are going to be available, all right? We can have it attached, or we can carry it in the blood as a dissolved gas. But now here's the problem. When we are at a tissue level where cells are located, and cells, after doing their job, if they make this CO2, if that CO2, when that CO2 gets to the bloodstream, and because I have water in my bloodstream.